Hi guys, welcome. Welcome, welcome, welcome. If you are joining me, if you have any questions, hey, I've done my in the book, but came up with question. Oh, you're very welcome, Alice. I'm sorry that you didn't find me sooner, but I'm glad that you have managed to find my resources helpful. So that's good to know. It's a long wait until you actually get to the point at which you get your results now, it feels. Oh, thank you for the share, Z. Thank you. I am currently working on my exchange and transport in animals, but I'm doing one specifically for mammals with my study guides because I realized one of the things that I do, which the textbooks don't tend to do, is I tend to put all the stuff together and make it link together in a way that makes it make sense, well, to me anyway. And so therefore I thought what makes it better would be if I were to make my study guide actually flow like you would expect if you were trying to learn the concepts of the transport system and exchange system in the animals. So I focused on the mammals for this one. I'm also currently working on the fish and the insects, but it takes a bit longer because obviously the girls are at home now and they are full of energy, which is great. Hi. Oh, thank you. This is my latest one. This is the one that I've literally just finished doing today. I've just been adding it in. I'm just about to add in the different parts to it to discuss what that actually is. But thank you. I've been working on a lot of diagrams lately, trying to make them look better. So the one that I was working on yesterday, let's give a little sneak peek of this was yesterday's diagram to show you the double circulatory system. So I want it to be that these diagrams are accurate, as accurate as I can make them, because obviously I'm using Illustrator, but also to make it so that it makes sense in a visual way. I'm very much a visual learner, and so therefore I need it to be that what I'm looking at makes sense to me, and so therefore hopefully makes sense to the reader as they read it and look at the different structures. So this is talking about the fact that the mammal circulatory system is obviously a double circulatory system, and the fact that the blood is entering both sides of the heart simultaneously. And that's the thing to remember whenever you're learning about biology, specifically A-level biology, you have to understand all the intricate parts, but the textbooks and the content that your teachers will be teaching you sometimes doesn't go into enough detail. For me anyway, that's how I find whenever I do my revision, whenever I am making my resources, I like to know every little bit and how does it interlink. So I like to see what's going on. And I'm trying to make these diagrams actually make sense visually, but also be accurate so that if you are studying for that A star, that you'll get that. Oh, thank you. So it's a difficult one to manage the content and the level to be high enough. What is the class about? Well, the class is basically, I've jumped on here. I do a live on a Wednesday, but this is going to be my last one for the summer holidays, unless I have a bit of time next week. But basically, if you have any questions relating to the content, I'm happy to discuss it. But I was just talking about my exchange and transport system in mammals that I have been working on for the revision guide. So if you have questions, if there's anything you want to learn or want to know, please do feel free to ask away. I do these sessions as a way of trying to support as much as I can. I'll be back every week at a Wednesday evening from September, but which exam board is best for A level? It depends. There is no right or wrong answer on that one. Which exam board do I like the most is a different question, but I would say there isn't one specifically. IB is fantastic for preparing you for university. So the International Baccalaureate is really good for that preparation because it feels like you're doing first year of university, the way that it examines it. OCR is an exam board for students who have more content to learn, but the questions are asked in much shorter. So you'll only get a 6 marker as a maximum number of points. With regards to AQA, it's got easier questions when it comes to the way it asks the questions until you get to that 25 marker on paper 3. That's when it becomes harder. So if you're an SE writer, then that's easier for you. At Excel, B is, I would say, more application based, but not as much content to learn. Okay, so what are the risks in cell culture if fungus grew way faster than my eukaryotic cells and how? What exactly are you trying to do? So if you're trying to culture cells and you are wanting to isolate those cells, you need to make sure that you are isolating the ones that you want. So you need to be sure in what you're actually culturing. But at the same time, if you're talking about fungus growing faster than the eukaryotic cells, then how are you actually sure? Because fungus is a eukaryotic cell. So how do you know the difference? How are you distinguishing between the different types of cells? So I'm trying to understand what you mean by that. Because eukaryotic cells are obviously your Protoctista, your fungi, your animalia, and your plants. And so therefore, that doesn't quite make sense. I think you mean your animal cells. You've tried to culture your animal cells. But if you're trying to culture them, you'd have to be sure that you have cells that can go through mitotic division. So, hi. So you're right, you can distinguish in the fact that fungus have a shorter cell cycle, but what you're trying to say is you said eukaryotic cells, your cells, and that you were culturing them. So I was trying to understand what exactly you're trying to do, but you're absolutely right. Fungus do have a shorter cell cycle, but it depends which organism you're actually looking at specifically, because the different fungi have different cycles. Hugo, I bet you're glad you've got no more biology exams. It's one of those things that it's hard to do all that work and then to do nothing, but at the same time, it's nice and refreshing to do nothing. Um, oh, thank you. Thank you. It's really kind of you. Oh, love from India. Thank you. That's really kind. Yeah, basically I just trying to help anybody who's interested in biology and also thinks in a different way, because there's a lot of content that you have to learn for biology. And if you can understand it, and basically, I look at this as being the next generation of scientists, the next generation of doctors, the next generation of um, students that I teach could go on and discover something which could change the way medicine's looked at. But unfortunately, sometimes the way our brains work hinders us in actually being able to achieve, even though we've got the creativity and the thought process. It's one of those things that can become quite difficult when you don't think or don't learn in exactly the same way as what's being taught to you. So I've been learning more about how my brain works and how I learn. And what I've been trying to do is then show that in my diagrams, show that in the way that I teach, and use that to help students see the bigger picture when it comes to biology. But it's one of those things, different people learn in different ways, and it's about figuring out what works for you, and finding the right way to learn that is going to help you succeed. So it's a hard one. Right now, I'm listening to a book called, oh, what's it actually called? Bright, Not Broken. That's what it's called, which is about the different ways to think about your brain and how you could be twice exceptional, but actually you might also have a disability that makes you hinder. Now, I've only just started the book, so I need to really read and carry on with that. But it has opened my eyes to think about the fact that we do look at the learning process very different. And sometimes we focus, specifically, I do anyway, focus on the negatives and focus on the weaknesses rather than focusing on our successes and focusing on the areas that we excel at. I spread myself very thin when it comes to doing different projects. I get really excited about something, but then I can lose interest quite easily. And it's only when I come back and re-engage with it do I actually see the success and the improvement that I've made. Oh, I don't know about that one. When you say they can't catch the azotes, what do you mean by that? And this is where I don't have all the knowledge when it comes to biology. I know the bits that I teach, I know the bits that I am confident and I would say an expert in, especially when it comes to, for example, OCRA. But if I was looking at something that I haven't covered in a while, then I couldn't tell you for sure on that one. So symbiosis is where you do have a relationship where it's a mutual relationship between both the something is benefiting and something is gaining, well, something, they're both benefiting in a mutualistic way. So symbiotic is the relationship, but I'm intrigued with this azote because I don't actually know that. And also I'm very tired. What do you think the grade boundary will be for the A level this year? I feel like I did put a video out about the different grade boundaries. I feel like they are going to be very similar to what they were last year. Do I teach immunology? I teach OCR and I teach the immunology part of OCR. Um, but it depends again which exam board you're actually studying as to what detail they go into. So if you're AQA, they go into way more detail on immunology with the assay side of things than they do on OCR. Um, oh, there we go. Azote is an old-fashioned word for nitrogen. Ah, so you're actually talking about nitrogen fixing. Yes, yes. And this is where, yes, I did. This is what I'm looking up because that's the thing of when you come up with a word, you'll use a word or your teacher maybe uses a word that I have never heard of before or I haven't used. And so therefore, because I'm not aware of it, I'm then straight away you get that feeling of your mind goes blank and you're like, oh, I'm under the pressure. I don't know it. What should I do? Well, actually, it's okay to say, I don't know. I don't know this, but I can go and have a look. And I did. So nitrogen fixing relationships that go on, then yes, that is a form of symbiosis. If we're talking about the rhizobium, which are the hi from Egypt, rhizobium is the bacterial cell that you the cells that you need to know that are inside the root nodule, they do have a symbiotic relationship. Exactly. And this is the thing that biology doesn't have. You're absolutely right. It's not just about the theory. It's about the theory that the exam board wants you to know. So it's important that you are looking at what the exam board that you're studying actually wants you to know and then going into the detail from there. And that's where a lot of students trip themselves up as well in that what they do is they try and find the style that works for them and they find a video that's amazing and it's really good and it's really insightful and it's got lots of information and then they find out that it's not actually for their exam board so they could have gone into way more detail than they need to, and it's one of the reasons why when I was going back to teaching A level again after doing IB for so many years, I had to go and look and see, well, what does OCR want you to know? What is the content that they're asking? And so what I then did was because I was going and looking for that knowledge, I couldn't find very much out there. And there were lots of YouTube channels for AQA, but for OCR, yes, there were a few that were out there, but nothing that gripped me in the same way that I would say for me to direct my students to, which is why I built my channel. Oh, thank you. Enjoy the rest of your evening. And if I'm not back on here next week, because I am going to take a bit of a break for the summer holidays, having the girls at home every day means that I'm a little bit more tired than I would be when I'm just focusing on work. So I'll try and jump on, but I might not, but I'll try and put some videos out when we definitely go and see the glorious sunshine. Unfortunately, the sun's not been out as much lately, but yes, thank you. Enjoy your evening. And I'm glad that you enjoy what I do. That's really kind. But I'm just going to be on here for a short time. I just thought I would jump on as I am still building my workbooks. I'm focusing so much on the workbooks and trying to get them done, but it takes a lot of time because I want to make sure, and this is one of the downsides of being a perfectionist, is that I want to get everything right. I want it to be accurate. I want it to help students. And so I don't want to miss anything out or to not give the information. But at the same time, how do you ensure that you're not giving too much information that it then becomes overwhelming? Such a fine balancing act. But yeah, I thought I would jump on here for a quick session, see if anybody had any questions they wanted to ask. Whilst I also still work through my booklet. So welcome if you are just joining me. If you have any questions, please do ask. Do you do GCSE bio as well? I don't tend to do as much GCSE bio now. The reason being is I have focused solely on A levels, and my timetable from September last year just got filled up with A level students. I did do a few GCSE sessions, and I can teach, and I have taught GCSE. More AQA than OCR when it comes to GCSE. I've done a few students. Oh, I have put some GCSE videos out for students. I did some chemistry and I did some biology GCSE, and if I have time, I will try and build some more sessions, but it all comes down to obviously having to do what is necessary to pay the bills, but at the same time, I want to help as many students as I can. But I've been focusing on the OCR A with regards to the A level. But yes, I do GCSE biology. And actually, it's important that I always look back and go back over the GCSE biology because I have to understand where students are coming from and what their struggles are. So I've marked GCSE for many years as well, because actually it's one of the things that you do as a teacher. You make sure you can see that transition going all the way through. 11 done with GCSE and is doing what would I say for GCSE students? I would say have a break, which is hard because, and I get that because I'm finding it quite difficult. You've gone from such an intense period of time where you have been working so hard on your GCSEs and you're about to start A levels, and A levels become quite challenging right from the get-go. So I'd say two things. Either take the time to actually enjoy the break because you're not going to get one over the next 2 years. But if you are the type of student who wants to get ahead, then what I would say is if you're doing OCRA, obviously check out my YouTube channel and watch my videos that I've got on there. But what I would say is start with a couple of topics, just having a quick look over, seeing where it's going to go, because that will help you in getting your mindset into a place where you're going to start the year as you mean to go on. There is a lot of content you have to learn for A-level biology. And so it's that whole thing of it can become very overwhelming very quickly. So I would definitely say have a break as much as you can. But if you get bored, then absolutely start reading, start looking at the biological molecules and it's interesting because a lot of students start with cell structure, and I would say cell structure, yeah, start with cell structure because that's where your teacher probably is going to start. But actually the one that's the most challenging and most interesting would be nucleic acids and nucleotides. So I would say look at that and look at the depth that you're going to go into when it comes to the likes of DNA replication and protein synthesis, because that's the one that so many students trip up over and have to do again and again and again. So it's, yeah, that would be my advice. Enjoy the summer as much as you can because 2 years will go. You'll be surprised how quickly you'll start and that it will just disappear. Oh, nice. You're doing the GCSEs in the winter. Are you doing the November exams? And is it there for IGCSE? If it's IGCSE, then do make sure you're looking at the practice exam questions that you can do. There's lots of stuff out there that you can do past papers, and I would always advise as many past papers as possible would be the way to go. But yeah, I just thought I would jump on here, and I am working through my revision study guide at the moment on the mammals and their exchange system. In fact, actually give me a second. I'll put a little sneak peek on here, and then anyone who actually watches my videos on here will get a sneak peek of what it's going to be like. So this is my study guide, which I am working on at the moment. So you can see it's about exchange surfaces, talking about how to measure them, talking about the information you need going into the mammalian exchange system. Then it goes on about the medulla oblongata and your different spirometer traces, how you read them, the alveoli, working your way through, hemoglobin, talking about the dissociation curve. It makes its way through to the different ways in which the carbon dioxide can be transported, your dissociation curve to do with Bohr shift. And then you've got your circulatory system, your different blood vessels, and then this last one that I'm currently working on, which is my tissue fluid formation. And that's what I'm doing right now. That's what I've been working through. And this will be available to purchase once I have finished it. But obviously, it takes time. It takes time to work on. So this is my one that I'm currently doing on Adobe Illustrator. And so this is what I was actually working on. And I was just making, and then I was changing the colors and doing, and that's been the biggest learning curve, is that I've been having to learn Adobe Illustrator at the same time as trying to do these revision guides. But I love it. I love learning new things. I love trying to see how I can change the different parts and watching other people's videos. Actually, that's what's been really fascinating, watching other YouTube creators and seeing how they make different structures on Illustrator. Someone's alarm going off. But yeah, that's basically what I do, or what I have been doing when I have the time. But right now, obviously, with it being the summer months, it means that I am now working on playing with my girls, enjoying the summer holidays as much as I can. Today we went on a bike ride, which was lovely, until my youngest fell. Oh, you're very welcome. You're very welcome. Um, Confident you got a good, oh, oh, I'm really pleased, and I hope you do get the grade that you want and need for whatever it is you move on to. Please do let me know when you get the results on, I want to say it's the 15th, I think it's actually the 14th. When is it? It's the 14th of August. When you get there on the 14th, please do let me know how you do. Yeah, 14th. So it's hard because I always think it's the 15th, but for some reason, I don't know why I'm feeling that the 15th should be a Thursday, but it's not, it's the 14th. Is Excel Biology one of the hardest exam boards for biology? I would say it's the hardest exam. It's a difficult one because I feel like some of the questions are very straightforward and some of the questions are really quite challenging. So it really just depends on the exam that you sit. But I do find that I actually really like the Edexcel questions because the way they ask them is challenging, but I like that. But that's more when it comes to graph and interpretation questions. When it comes to just the science and them checking your knowledge, I actually would say they're quite straightforward, the questions in EdXLB. From the ones that I've seen anyway, I would definitely say that's the case. But yeah. That is what I've been working on. Okay, if it's the building close by us that's going off. Tends to be the way. It's an empty building, and unfortunately, someone gets too close to it and the alarm goes off. And then we get the joyous noise, which is very distracting. Oh, there we go. It's off now. Do I think it's possible to learn all of the bio EdXLA in 1 year? You've just finished year 13. So if you've just finished year 13, did you do a different exam board and now you want to try EdXLA? What exactly? Do I think you can do biology in 1 year if you are doing an accelerated course? Yes, I've taught accelerated course a number of times. Are you a retake or are you someone who is changing the exam board? So there's lots of different questions I would want to understand. Do I think it's possible? Yes. Do you have to commit? Yes. Do you need to ensure that you have the time and headspace to do that? Yes. But absolutely it's possible. It's about how much effort you put into doing something. But anything is possible if you have the time, the motivation, and the right resources, I would say. Okay, so you're self-teaching. Which exam board? Is it OCRA? If you're self-teaching, the key thing you need to make sure that you don't get caught up in, AQA. Okay, so lots of practice, lots of past papers looking at the mark schemes. AQA's mark schemes are actually really good to use and know exactly what they want you to know. I would say the hardest thing for you is paper 3 because of that 25 marker and the mark schemes don't really tell you anything. Obviously, when you do the 25 marker, they're looking for at least you doing connections with 4 modules minimum. So the key thing is lots of practice of interlinking of the topic areas. So seeing how, for example, if you were looking at cells and you're looking at the different structures inside cells, can you connect that to hormones being produced? And then can you connect that to movement of substances across a membrane, for example? So you're not a retaker. You haven't done bio since GCSE. You just struggle with picking what to do for uni for a bit. Oh, okay. So you want to take a year out and that's a great thing to do. If you're unsure what to do, to get a flavor for lots of different things. So yes, I would say that it's possible. What did you do for your A levels, if you don't mind me asking, the ones that you currently did? What did you actually do for those A levels? Would be my first question.